Okay, welcome back to part 2 of chapter 12, Control of Gene Expression. So with all those modifications we talked about in the previous part, okay, which can affect chromosome structure, multiple of them can happen at the same time to turn a gene on or off. So here you have example where you have a gene that is switched on because you have active or open chromatin structure where you have stretches of your DNA molecule that's exposed to any protein or the initiation transcription apparatus to bind, okay? And you have unmethylated cytosine, which is no DNA methylation, okay? So these open circles represents unmethylated C's or your cytosine, and then you also have acetylation of the histone itself. Okay, so these histone tails have been acetylated so that it really destabilizes that chromatin structure to open up these areas for gene activation. Okay, so a combination of no methylation on the DNA and acetylated histones can come into play to turn a gene on. Now, to turn a gene off, the opposite happens. We're silencing the particular genes involved by making that, you can see, closing of the gaps between histones so that DNA is more tightly wrapped around the histones. So these usually involve methylated cytosine, which are represented by these red circles. So these are methylated C's, which we were talking about as your heavily methylated gene. And then you have deacetylated histones, so that's why you don't have a tail coming out, because that tail is tightly wrapping around that DNA molecule, as we have seen earlier. So when there's no acetylation of the histone tail, those tail would, instead of raising their hand up like this, okay, they will be tightly wrapped around your DNA molecules to prevent those DNA molecules from falling off or uncoiling. Okay, so both of these cases can have an additive effect on each other to really protect this portion of the DNA that will turn genes off. Okay. All right. So what about changes at the transcription level? What can affect gene expression there? Remember that from our transcription chapter, we learned that many protein factors have to bind to the promoter sequence of DNA in order to initiate transcription. Well, it turns out that there are two parts to the promoter region. Well, just like how we have structural gene versus regulatory gene, we have two different promoters. One is called the core promoter. The other is called the regulatory promoter, okay? Those proteins that bind to core promoter is called general transcription factor. Okay, they bind to the core promoter region, including something called the Tata box. Okay, we didn't talk about this when we were in that chapter, but Tata box is actually a critical region that's, of course, with the sequence TATA, nucleotide sequence TATA, within the core promoter. Okay, so those general transcription factors, along with your RNA polymerase, will assemble on the core promoter to make what's called the basal transcription apparatus. This basal transcription apparatus, though, will only allow minimal levels of transcription. You need something else more to turn that gene even more so on or express at a higher level. There are proteins called transcriptional regulator proteins that can bind to the regulatory promoter region that can either activate or repress gene expression depending what binds, okay? So that's why we have a core promoter region and regulatory promoter region right there, okay? And the core promoter region is for the Tata box, where your basic stuff that initiate transcription like RNA polymerase and other, you see all these circles all over here, those are like little factors that we didn't talk about because there's enough stuff for you to memorize already, but doesn't mean they don't exist, okay? There's actually many multiple parts of the transcription apparatus that must all combine and bind together to assemble this basal transcription apparatus on the core promoter. Now again, this core promoter with the basal transcription apparatus will only transcribe at a minimal level. In order to really turn this gene on or express a lot of the proteins, you need the help of transcriptional activator proteins. So those transcriptional regulator proteins can have activator or repressor. If an activator binds to the regulatory promoter, then it will stimulate transcription by either stabilizing the basal transcription apparatus and or indirectly stimulate transcription through a co-activator. Okay, so co-activator is something that might bind to your transcriptional activator, which will connect to your basal transcription apparatus through another thing, maybe mediator. Okay, but I'm just not adding all this stuff. Some of this stuff I'm not talking about because it's just a lot of things for you to know. But activators and co-activators work together to either stabilize this basal apparatus or make this more activated, okay? Now, repressors are proteins that can inhibit transcription by either competing with the activator for the same binding site, like the regulatory promoter, or it can bind to a site close to the regulatory promoter that can inhibit activators from binding, or they can ultimately interfere with the assembly of the basal transcription apparatus itself. So remember, here you have your transcriptional activator. But what if you have a repressor binding right here? Well, if a repressor was to bind right there, then here comes activator and it can't bind, okay? So if activator can't bind, being blocked by the repressor, then it cannot enhance that gene expression, okay? So that's how repressor works. Sometimes it can bind at the exact site, or it can bind somewhere close, like maybe here or somewhere close to it, and it will block somehow from the activator protein from binding to their favorite site on the regulatory promoter part, okay? Now, this part I'll talk about in the next slide when we get to enhancer, so don't worry about this yet, but this is essentially how activators are helping with transcription, okay? It's by stabilizing this basal apparatus or turning this gene on more often, okay? Now, within the regulatory promoters, there are different consensus sequences that can exist for different activators and repressors to bind, okay? So remember, we talked about consensus sequence. Those are sequences that are very much alike with each other. And they're specific for different types of whether you have activator or repressor. They all can be mixed and matched into different combinations of binding sites for different promoters. Okay, so hence here you have 3 different promoters, okay? And each promoter can be regulated by its own unique combination of activators and repressor for different genes. For example, here you have GCs as your consensus sequence, but here in this promoter, you have a GC too, okay? CAAT is another consensus sequence for something else, okay? So all of those things can be, combined or recombined for different activator or repressors to bind at that particular promoter, but they're all unique by promoter sequences, okay? Or for different genes. So here comes the enhancer part. Okay, so enhancers, like how I was talking about, are not proteins. They are regulatory elements. So they're not regulatory gene, okay? They are regulatory element. They don't encode anything. They don't get transcribed, okay? They are just sequences within the DNA that sits there, okay, and they can affect transcription at distant genes by stimulating any promoters in the vicinity or close by allowing some stuff to bind, okay, because they're just sequences that are just sitting there, and hence something can bind to it that can enhance the effect, okay. Usually a regulator protein must bind to the enhancer, okay, some regulator protein, maybe activator, okay, in order for the gene to be activated and stimulate transcription. So, for example, an activator protein can bind to the enhancer site. So, here's that other part I was talking about that I wasn't talking about in the last slide. So, this section, you have enhancer separated by some stretch of DNA, and then another regulatory promoter region, core promoter region, and then you have your actual gene, okay? So this enhancer is not really part of the gene whatsoever, neither is it part of the promoter. It's just some other sequence out here, but it can bind to activator protein, and somehow this activator protein likes this particular mediator, or it likes the RNA polymerase. And this overall, along with this activator protein here, can both contribute in stabilizing this structure and allow for activation of this gene transcription. Okay, so that's how enhancer is able to provide an extra site that will, in a sense, enhance the expression of that particular gene. Okay, if you have enhancer, of course, you have the opposite, which is insulators. So insulators, on the other hand, are sequences that allow proteins to bind, but it will block the effects of enhancer. Okay, that's why it's called insulator. It's like it's insulating the effects of enhancer with the genes that are in the vicinity. Okay, and this can differ by the position of the insulator with respect to the enhancer. Okay, so for example, you have an insulator right here that is between the enhancer, okay, and the promoter, okay? So this enhancer can act on this promoter because there's an insulator in between there. Remember, enhancer can act on anything close by. It doesn't have to be a particular gene, because right now you have gene A here and gene B here, okay? There are two enhancers separated by insulator. So enhancer 1 cannot play a role in stimulating transcription of gene B because of this insulator. Okay, but if the insulator is lying outside the region between enhancer 1 and this particular promoter, then this enhancer will enhance gene A's expression. Okay, so this insulator has no effect between this promoter and this enhancer 1. Neither does the insulator has an effect on enhancer 2 and this promoter, okay? All this insulator's blocking is from this enhancer 2 to act on gene A's promoter, and for enhancer 1 to act on gene B promoter. Okay, so that's what that insulator is blocking from. Okay, and just like enhancer usually requires some kind of insulator binding protein to block the enhancers from working. Rarely do we have sequences of stuff that is just hanging around doing whatever function it has on its own. It requires some other proteins to bind usually. Okay. All right, so for controlling gene expression at the mRNA processing step level, we have already talked a bit about this, so I'm just going to go quickly through this, okay? Remember, we talked about the various factors that are added, including the 5' cap, which is this thing right here, and then you have your poly A tail at the 3' end of the mRNA molecule. Both of these play a role in the stability of the mRNA, even loops the mRNA back to stabilize the position of the ribosome and enhance the start of translation. Okay, so that's why these two things are very important. There's particular proteins called poly-A binding proteins known as PAPs. Okay, they bind to 3' poly-A tail, and it actually plays a role in helping the stability or protection of the 5' cap. Okay, but scientists found that if they were to chop this poly-A tail off to a very, very short critical limit, okay, the 5' cap actually gets removed somehow, and mRNA molecule gets degraded. So they know for sure that a certain length of this poly-A tail is very critical to stabilize this ribosome on the mRNA, and this particular yellow thing here actually stabilizes the 5' cap. Okay, so when you remove just the poly-A tail itself, the 5' cap somehow also gets removed. Okay, so that's why these two things are very critical for the stability of the mRNA, and hence it controls or play a role in how often that gene is expressed. Okay. But with the details of how alternative splicing is regulated, okay, we won't talk about that. But just to remind you, alternative splicing is the other way we can modify how a gene is expressed, okay? Remember with alternative splicing, what is that? Okay, you have exons and introns, okay, and after transcription you can have both exons and introns in there, but through mRNA processing you're going to remove all of those introns, but whether those exons all come together in one finished mRNA molecule can differ. For example, one mRNA molecule can have exon 1, 2, and 3, or another mRNA molecule can just have exon 1 and 3, or another possibility is another mRNA molecule with just exon 2 and 3. Okay? So this is what going to generate different proteins in different tissues at different times of development, just by that one mRNA molecule from that one gene. They also say that the regulation of splicing can also gives you means of controlling the gene expression. Okay, but we're not going to dive deep into how it's regulated. When I looked it up online with research articles, many say that the details of how alternative splicing work is very complex, okay, and how they regulate it is very complex. So we won't talk about it here, and actually some research is still going on to how to elucidate and find out more details about exactly how different genes alternative splicing work. It doesn't work just one way for a million genes. There's different ways of how alternative splicing work for different genes. How does that particular gene know to just take exon 1 and exon 3? How does that other gene know to go all the way through 8 exons? That is still the question, okay? All right, the last modification we will discuss in this lecture is controlling gene expression by using RNA interference or RNAi molecules such as microRNAs and small interfering RNAs. Now, to destabilize DNA or both of them, okay, because here you have microRNA and siRNA. Different ways of using this can either end up with destabilization of DNA or inhibit transcription or translation, okay? What we know about RNAi is that they're usually in the realms of 21 to 25 nucleotides long, so they're not very long molecule. They're short little RNA molecules, okay? And how they're formed is actually from originally a double-stranded RNA. and they will be cleaved by an enzyme called dicer, okay, right here. So dicer is going to go in there and chop this double-stranded DNA up, okay, into smaller pieces like these, okay, and these smaller pieces then will now become single-stranded RNAs, okay, very small fragments of them. They will then bind to other proteins to form what's called the RNA-induced silencing complex, or the RISC, okay? Now, this is where the RNA component will then pair and bind to its complementary sequence on its targeted mRNA molecule. Usually, where they bind is kind of close by the 3' UTR region. So in this case, you have that short little siRNA here binding, which part of that RISC complex. And both of these will then be targeted for that particular mRNA molecule. Let's say this particular sequence was derived from the same sequence of this mRNA. So that's why it binds perfectly with each other, binds perfectly there. So by having this whole bunch of protein in that area, it's kind of acting like a repressor protein, okay? Where then it blocks other stuff from coming on, like translation, and so no translation can be made. And with siRNA, it can actually lead to degradation of the mRNA molecule. Now, there are 3 different effects that can happen with these RNAi molecules. For siRNA, which is your short or small interfering RNA, they will bind with proteins, again, to form your RISC complex. RNA component of siRNA will pair perfectly, okay? They will pair perfectly with the complementary sequence on the targeted mRNA molecule. This will then activate another enzyme called slicer, okay? And slicer is actually going to cut at the mRNA site at the location in the middle of that siRNA sequence. So let's say this is getting messy here. Let me erase that. Okay, so with that, you have your siRNA right there, and the middle of that sequence is where slicer is going to know where to cut. So it's going to make a slice right there, break apart the mRNA molecule initially into two pieces, okay, 2 bigger pieces. This will trigger somehow further degradation of the rest of the mRNA molecule into smaller pieces, and hence it can't be transcribed once the mRNA molecule just in small little fragments everywhere, okay, and then it will be recycled as nucleotides for other cases, okay. So that's how siRNA works in degrading your mRNA molecule, therefore preventing translation from happening. Again, it reacts at the translation level. Transcription still happen. You have your mRNA there, but the mRNA itself is going to be triggered for degradation once an siRNA along with RISC complex bind together. Okay? All right, so microRNA, mRNA, are made in a similar process with dicer, cutting that double-stranded RNA into smaller pieces, fragments. As you can see, here's dicer, and it's slicing that big double-stranded piece up into smaller pieces, okay, or smaller, shorter single-stranded fragments. And once microRNA forms a RISC complex, though, the RNA component actually pairs with the mRNA sequence imperfectly. Okay, imperfect is the key word there when we think about the difference between siRNA or and microRNA. That's the major difference between the two. siRNA is very specific and targeted for that specific mRNA sequence, whereas microRNA can actually not be very specific, and that's why it binds imperfectly with the mRNA molecule, because one microRNA can actually bind to multiple different sequences of mRNA molecule, okay? So siRNA pairs perfectly, microRNA pairs imperfectly, but either case, the binding of this mRNA with the RIS complex with microRNA will still inhibit translation. Okay, we're not going to go over the details as to what steps is here to get to the translation, inhibition of translation, but ultimately it will also lead to blocking or inhibition of translation. The third effect that RNAi have on mRNA, well, not mRNA molecule in this case, it actually affects the transcription level. Okay, so RNAi react going with some siRNA. Okay, so only some, not all siRNA works this way, but some siRNA molecules can combine with proteins to form a different complex called the RNA-induced transcriptional silencing complex. Okay, so it's not RISC, it's RITS. Okay, RITS complex, not RISC, not that one. That one is for degradation or inhibition of translation by either your siRNA or microRNA. RITS is where your siRNA is binding with the protein complex. That's not going to affect the mRNA. It will actually affect complementary sequences in the DNA molecule itself, okay? Or even as the RNA molecule is being made during transcription to repress the process, okay? So what it does is that it actually blocks transcription by attracting enzymes like this big orange circle here that will methylate the tails of the histone within the DNA sequence right there, okay? So remember we talked about histone proteins. If it becomes methylated, depending on the case, in this particular scenario, the histone methylation ends up making the DNA wrap or coil around the DNA coils around the histone even more tightly than it was. So it ends up restricting the access of proteins and enzyme that's necessary for transcription to occur. So once that happens, that methylating enzyme comes in here, methylate histone or methylate the DNA itself, either way. So that's why it says methylate DNA or histone. Either way, it will inhibit transcription, okay? So if it inhibits or methylate DNA, we already know methylated DNA will result in gene repression, okay? If it methylates a histone, in this case, it will make the DNA coil tighter around those histone, and hence it both ends up inhibition or blocking transcription from happening, okay? So both siRNA and microRNA mix the RISC complex through dicer and slicer, okay? And that will inhibit translation, whereas some siRNA can make the RITS complex, which will then affect transcription and block transcription from happening, okay? So that's why I said this chapter is pretty short, okay? It concludes chapter 12 here. Usually it's a very long chapter in the textbook, but we are only focusing on mechanisms of controlling gene expression in eukaryotic cells. So don't forget to study for the next lecture exam, okay? Which will cover all the way from chapter 6 through chapter 12. All of these chapters are actually very straightforward, as you can see, compared with the last 5 chapters, because there's not so much calculations and problem solving involved, but there are many steps and processes and a gazillion of names of different enzymes that you have to know and be 100% clear about in order to make sure you're not confusing yourself on the exam between mechanisms and processes, which enzyme is for what step goes with what blocks this process, etc. Okay. The other thing is, don't forget those scientific experiments that we mentioned for proving DNA as genetic material. And there's random experiments here and there to prove how translation works, etc. So it's a lot of terms to remember. So make sure you start making diagrams, note cards, whatever it is that will help you be very 100% clear of what goes with what chapter, okay, or what processes, okay? All right, so let's go through the practice question. Okay, so the kind of gene whose products interact with other nucleotide sequences to affect their transcription or translation is a regulatory gene. Okay, the one that's actually encodes the protein that does its job is called structural protein. So those that interact with other nucleotide sequences to regulate gene expression is regulatory gene. Okay, and what constitutive gene are those that are constantly turned on. You don't turn off those genes. Those are genes just on all the time because they're essential genes. Okay, for example, remember I mentioned ribosomes because we always need them. They don't need to be turned off. That's why they're called constitutive gene. Okay. All right. Transcriptional regulatory sequences in the DNA include all of the following except the mediators. Okay, we didn't talk about mediators. We talked about core promoter, regulatory promoter, enhancer, insulators. Now, the other thing to think about is this is talking about regulatory sequences of the DNA, not regulatory proteins. Okay, it's not saying it's proteins, it's sequences, which means these are just sequences within the DNA, not proteins that binds to the DNA. And all of these are sequences within the DNA. Core promoter initiates the process of transcription by allowing your transcription factors and basal transcription apparatus to bind. Regular promoter is what allows activators or repressors to bind. Enhancers, again, is for your activators to bind. Insulator actually blocks the effects of enhancers. Okay, so all of these has to do with regulatory sequences. Okay, but mediator is actually a piece of protein. Okay, a glob of protein. So what is the role of insulators in controlling gene expression? They block the role of enhancer. Okay, that's what it is. Okay, that is pretty simple. Which two mRNA structures interact with each other to provide mRNA stability? You have the 5' cap and 3' poly A tail. Okay, so these two, remember that you have your 5' cap, okay, and then you have your 3' poly A tail that has these proteins associated with it that binds to your ribosomes to stabilize this translation complex, and then it can, sort of go ahead and translate this mRNA molecule to protein. All right, so in eukaryotes, there is a variety of post-translational mechanisms of gene regulation. Which of these mechanisms acts in the nucleus? The key word there is nucleus. So alternative splicing is the only thing that acts in the nucleus. Here it says translation, which means cytoplasm. mRNA-mediated translational repression, cytoplasm. Addition of phosphate or sugar groups to proteins, which means this is talking about post-translation, translational stuff, which means it's in the cytoplasm, okay? So siRNA and microRNA inhibit gene expression by all of the following except by stabilizing translation machinery. It does none of that. Okay, if it's stabilized translation, then it's not doing what it's supposed to do. siRNA and microRNA is to stop translation from happening, destabilize the translational structure, or chew up that mRNA molecule, either way, or even inhibit transcription with the Ritz complex that we talked about. But it does not stabilize translation machinery. When histones are methylated, which of the following occur? Both A and B occur. Remember, histones are methylated, so methyl groups are added, which is CH3 groups, and repression or activation can happen. Remember, because it depends on the case, okay? It's a case-to-case scenario basis whether histone methylation can lead to activation or repression. It is only acetylation that only gives you repression of the gene. Activation of the gene. Okay, so acetylation, activation of the gene. Okay, A. So that's another easy way to remember. Acetylation always gives you activation of the gene. Heavily methylated DNA is associated with repression of transcription, because this is talking about methylated DNA, not histones. Okay, so methylated DNA always represses transcription because it will lead to tightly coiling of the DNA around the histone proteins. Okay, and remember that methylation of DNA usually happens on the cytosine. Okay. Which of the following is a difference between small interfering RNA and microRNA? Your siRNA tend to base pair perfectly with their targeted mRNA sequence, whereas microRNAs often pair imperfectly with the mRNA sequence. Okay. Which of the following changes in DNA and chromatin structure are known to play a role in the regulation of gene expression patterns that lead to differentiation of cells and cell specificity in a sense? DNA methylation and histone modification. Those are the type of things we've been talking about that can happen in a cell. Okay. All right, and that's all. So this wraps up chapter 12. Remember to study for your upcoming unit exam on Friday, lecture exam on Friday that covers the 6 chapters. And let me know if you have any questions. See you.